Our investigation into this Goss's wilt started in 2009. Previously, it had been a mild, weak disease in 6 counties in eastern Colorado, western Nebraska. Then all of a sudden, in 2009, it jumped clear to Pennsylvania and north to Manitoba, until right now it affects about 80% of the U.S. corn crop.
Standing in front of a field that we stopped at 3 weeks ago, and what we're looking at were disease stalks and shanks that were rotten. Now at that time it was early phase of the disease, and we know how it works is the bacteria and the semi-metallic crystal end up plugging the plumbing tissue of the plant. This is what we're seeing in about 90% of the acres in Iowa and much of the Midwest. These are fields that all should be dark green, yet on September 16th, instead, everything is brown. This is a new bacterial disease that's killed the corn early the last 8 years, trimming yields, raising the threat of having contaminated grain being harvested from the fields. The National Ag Statistics Bureau came out with their estimate last week, and they said 26% of the corn in Iowa had reached maturity. 90% or better was dead. So we have to make the assumption that roughly 70% died before it reached maturity. That is not normal.
Yeah, it's amazing. This corn was bright green Labor Day weekend on Sunday. I was here. I came back 2 days later and it was all brown. And that's when I realized that the Empruv is really doing something on mine.
What do you see for your size?
Disappointing.
There's an untreated ear. You lose the green tissue, you lose the ability to fill the kernels. And also
these are going to be a lot smaller. And this stuff, let's see, we were looking at black layer on the other one. I'll bet this is, oh, look at this. Kernel depth's really shallow.
About 3/8 of an inch, so you're almost a 1/4 inch shorter.
Died before its time. Look at that.
Ooh. See, the clavibacter rots all the tissue, dissolves the woody tissue. Cobs are mushy, you won't be able to get them off decently. And separating the grain from the cob is really tough. Grain quality, that's the stuff you don't want to put in your bin. It's the new Goss's. Reason why is the actual Goss's is caused by a clavibacter bacteria involving a semi-metallic crystal, and it stays in the soil. Is probably being transmitted either by the seed or through the air, because we're picking it up in combine cab filters and vacuum, grain vac filters.
We have a number of new diseases and also many re-emerging diseases that on the Threat Pathogens Committee for the United States I've served on for about 40 years. We look at these re-emerging diseases as diseases that used to be controlled very effectively, or that we knew were there kind of sitting in the sidelines, but they were never of economic importance for us to get excited about. Those diseases now are becoming very serious and threaten the sustainability of our agricultural production. One of them that I'd like to discuss is Goss's wilt on corn. Goss's wilt as an economic problem for us is an induced disease brought to most parts of North America and the rest of the world through genetic engineering. Now, I mention that because there are 3 things that you have to have for a severe disease. You have to have a pathogen. In this case, the bacterial pathogen is Clavibacter Michiganense nebraskaensis. You have to have susceptible corn hybrids, and that's what's been brought to us by genetic engineering, because in order to have expression of the genetically engineered traits, they had to go back to some of the more susceptible corn hybrids. You also had to have an induced or a favorable environment for expression of the pathogen in those susceptible corn hybrids. And that's because when you apply glyphosate or with the genetic disruption of the genetic code, we see an increased susceptibility to many diseases that are requiring fungicide application. When we apply that fungicide, that you nullify 6 of the 7 genetic characteristics for resistance to Goss's wilt. So we provide a more susceptible plant, we change the environment favorable for the pathogen, and we have a more pathogenic and increased virulence in our bacterial pathogen. Goss's wilt was formerly considered a very wimpy disease. We now see it with that induced intransigence, so that it costs us 1+ billion bushels of corn annually in the Midwest anytime we have a favorable environment for this disease to express it. Goss's wilt is indigenous to North America. It's a disease that originated here. It's now been spread throughout the world primarily through seed stalks. It can be seed-borne, waterborne, and wind-borne. It can also be bug-borne. We also see a reduced nutrient density in our crops, so that it's a loss that we don't attribute a great deal to, but it has a tremendous impact when it comes to feeding our animals or to feeding our human population. And this is a vascular disease, develops systemically. That critical period where you have a higher nutrient need is one that is especially reduced as a result of the vascular plugging from this pathogen. On the leaves, you'll see the lesions then will follow the vascular architecture of the host of the plant, and so you get long streaks of water-soaked lesions that will develop. They'll start from just a small spot, and then it spreads from those through the vascular system of the plant. You'll also see what we call a salting out of the bacteria. The bacteria is able to multiply and grow to the extent that it actually just seeps out of the tissue and appears as a salt-like deposition on the outside of the tissue, and we call that a salting out effect, a typical characteristic of the Clavibacter nebraskaensis pathogen, in this case the bacterial pathogen causing this disease. This then causes the plants to die or to prematurely age. These are symptoms essentially of just starvation. They're denied the water and the nutrients required to keep those tissues alive, and so you see the effects then overall on the plant. So it dies before maturity. That latter 30 days of maturity is very critical for nutrient density. That's when your protein, when your grain fill is maximized, and that's the period that's cut short with this particular disease. So that if you just look at the surfactants in the fungicides or in the Roundup and other herbicides that are used, those surfactants are there for a purpose, and that purpose is to increase penetration of the pathogen that the surfactant alone will eliminate or nullify 6 of the 7 genetic sources of resistance to Goss's wilt. When the glyphosate is applied, it nullifies the 7th source because it shuts down those physiological defenses that the plant has, even though we have genetic pathways that have been inserted to restore part of the shikimate pathway. They're not fully effective in restoring the resistance to Goss's wilt. A normal, non-infected corn plant, you'll see the ears dry down and then droop and drop because of the weight of that kernel as it's fully developing and storing all of those nutrients for us. What we see with Goss's wilt is that you'll see the top part of the plant, that highly efficient photosynthetic part of the plant at the late stage of development and grain fill stage, that actually dries off and dies because the vascular system has been plugged and it's no longer getting the moisture and the nutrients that it requires. Other corn hybrids and other situations may die from the bottom up. And again, this depends on the environment and stage of infection, stage of disease development. So we see the 2 conditions. Usually with Goss's wilt, then the whole plant will finally die off 2 to 6 weeks before genetic maturity is established. So that when we focus on the pathogen with Goss's wilt, we need to focus on those things that will reduce its virulence. That becomes a nutritional factor. The biological control has one of the greatest potentials for controlling Goss's wilt, so that biological control with an organism that can compete and can suppress the activity of the clavibacter becomes a very viable option for us. And one of those that we're seeing with some very excellent success has been the Empruv type of an approach.
Well, my name is Salam Awada, and I'm the president of AgSciTech Inc. So what is Empruv? Empruv contains a spectrum of ingredients. The main components are biological extracts or biochemicals. There is also some nutrients, as well as buffers, stabilizers, penetrants, natural growth stimulants, and natural surfactants. Some of the components in the biological extracts in Empruv activate the natural defense or immune system of the plants to help protect it against infection by pathogens such as bacteria and fungi. These components act as elicitors by activating the natural defense system of the plant. There is also stabilizers because we know as we apply Empruv, we sometimes mix it with other products. So it's critical to have stabilizers to prevent the components from reacting in the mixing tank with other ingredients. Also to help from interaction with components in the soil or within the plant itself. Empruv is not a bactericide or fungicide. Empruv boosts the natural defense system of the plants and they prepare the plants to better fight stress and pathogens. We are excited about Empruv. It's a new product in our line of products. It's exciting because it's targeting a new technology, which is the defense system of the plants. We're excited about what we're seeing also in the Midwest. I've seen a video and I've seen pictures of on actual fields that have been holding green until October. So that's always makes us proud and happy with what we put together and what we work on, because Excitech is dedicated to research, and we're happy to see good things happening that helps the farmer.
I'm standing in another healthy field in northern Story County. Again, the plants are green and healthy, even in spite of having the same weather, same temperature, same amount of rain as other fields. Yields here versus other fields, he's probably going to be 40 to 60 bushel an acre better. Proof is in the pudding. I had this here in my hand earlier. I just walked in, grabbed another average ear. Diameter on these ears is phenomenal. Last year, this field was running 265 to 2,800 bushels an acre. To draw a comparison, this field, we're tickle pink with the yield, standability is good, plant health is good. Right next door is a field that was hit very hard by the disease. I'll stand in front of it. You can see it's completely brown and extremely dead. A person might be wondering, why are we so concerned about having a healthy plant with green ears? The answer is, what the leaves are doing on the plant is converting sunlight energy, carbon dioxide from the air, and water from the soil into sugars, and the sugars make up most of what fills the grain. This would be coming from the green field. Now from the brown field where the leaves were brown, it died early, we've got ears that are looking just like this. This was picked from one of these fields. Guess which field?
I've had a lot of disease in my other fields for years around here. We're in a lower hanging area that gets a lot of late season disease. Usually around Labor Day weekend, the plants all pretty well turn brown and die prematurely. So that's why I was very intrigued with the Empruv product that Bob had told me that he wanted me to try. So we put it on right at tassel. I've been really amazed compared to the fields around my neighborhood, and here we are today, and this is 113-day corn that was planted on the 6th of May. It's not black layered yet, and we've got green plants all the way top to bottom. As you can see, we're filled out and still adding to the yield at this point in time, which is very unique for the whole Midwest region. I travel 14 states. In the last 3 weeks, I haven't seen anything but brown corn everywhere I've went.
So farmers have to be aware of it. They need to educate themselves on it. Proper nutrition and having a high enough micronutrient and nutritional level in the plant to help the plant mount an immune system response is what they have to do. And then the Empruv is probably one of the major tools in the toolbox on it. But compare the ears. Is that
so you got a choice.
Yeah. This one
come from an adjoining field.
This one or this one.
Yeah, these are all Empruv.
Shallow dent, shiny. Seeing is believing.
Certainly making a believer out of me.
The corn on corn with the Empruv on was yielding 275 to 295 bushel an acre. The best he's ever had before was 225. Looking at the ear here, it is filled right to the tip, kernels up and over. Go to the one right to its right, same thing.
Okay, well, we're going to measure ground corn samples, and we're looking at the difference between the use of Empruv and then corn that's not being treated with Empruv for changes to Goss's wilt. And so what I'm doing right now, I'm going to use this, which is the Bruker SD3 XRF. And what that means is X-ray fluorescent technology. So we're going to use X-rays, and we're going to energize the atoms in this sample that I'm going to put on there. And this is ground corn kernels. This is our BioImprove at the D level. And then we're just going to press it down because we want to take the air out, because this instrument is so sensitive that we'll actually measure the photons of argon in the sample if we don't just sort of try and make it a little tighter and give it a little bit more density. So we just press it down with a spoon. All we've done is ground dried corn in a coffee grinder. And now we're going to measure it, and it's this simple. This instrument is calibrated for food grains for both the micronutrients and the macronutrients. This is our rhodium peak here. Now the tube itself is made out of rhodium. The reason rhodium was chosen is because most plants don't ever take up rhodium. In fact, rhodium is a very hard element to actually get access to. So it seemed a good idea to make sure we're using rhodium instead of some other element that we might actually want to look at and would be overlapping. Now, again, this is our rhodium peak, so if we want to make sure our matrix is the same, we need to have an overlap on the rhodium peak. But let's look at what so this is our potassium, this is our calcium here, sulfur, we've got phosphorus next, and we've got silica down here and aluminum. So now let's go to concentration. We see silicon, phosphorus, sulfur, potassium, and calcium. And this, I remind you, is the BioImprove middle of the cob kernels. We're going to move A to B, and our spectrum is going to change color. And now the non-treated D kernels, and I'm going to press it down again. Okay, and we're going to run this next sample. And now you'll see a red line start coming up. This is our non-treated sample, and we're comparing it to our BioImprove sample at the D level, or the middle of the cob. So we can see our matrix is fine. We can compare. Now this is interesting because you can see that our non-treated cob from the Goss's Wilt is actually lower in almost everything. So it's lower potassium, lower calcium, lower sulfur, lower phosphorus, but actually it looks like it's about the same silicon. So why don't we look now? This is where we're doing our macro elements. So let's just compare. Well, our silicon is actually quite similar. And we look at the phosphorus, and oh my gosh, look at this. 1300 to 303, 1200 sulfur to 700, 3900 potassium to 2200 potassium, and then the calcium, well, calcium is not nearly as affected as all the other elements. This whole cob, if we look at the numbers here and we compare them, this whole cob of corn is nutrient poor. It's a poor food source. It's poor for animals, it's poor for people. This is much better, and these are the kinds of numbers that we want to see when we're looking at food density. So, better numbers, poorer numbers, the effects of disease on translocation and translocation into the seed head. Now, here's the other thing with that. When a seed is germinating, it needs these nutrients. That's why those nutrients are in the seed, because the seed actually needs them in order to grow a plant. If we were growing this for seed, we now have a problem. These seedlings will lack vigor because they don't have enough nutrient to actually grow a vigorous plant. The other thing about this is that our non-treated plants actually die and stop filling anywhere up to 4 weeks before our treated plants. Any plant that is severely stressed by disease knows that stress and goes immediately into reproductive mode and tries to get as many viable seeds as it can. But it's forcing the issue. So that's another reason why we see this decline in nutrients, because it doesn't have, and because its translocation system, the way it moves the nutrients up into the plant from the roots, it's all blocked up because that fungus, that bacteria, is actually in there. And the thing is, when we get these lesions, not only do we get the one disease, but we'll get other diseases, and the whole system will become blocked. And so it's like we have this block in the tube. So if you think about it like a hose, it's like you've kinked the hose and nothing's going up. Now we'll find some other ways up and some other ways down, but they won't be nearly as good. And when we're forcing the issue, because we're all stressed, we need to reproduce as a plant, it means that we just take what we've got and we put it in there and hope for the best.
What myself and other members of our group have discovered through 8 years of working with this new Goss's wilt disease is that we have to use a multi-step program. There's no silver bullet. We first of all have to improve the health of the soil through better cultural techniques, use of cover crops, build the soil biology and understand it. Next, we need to build the nutritional level of the plant as high as possible. And lastly, the product we're looking for was something that would knock out the Goss's wilt on a long-term basis through the season. We found it. It was the Empruv. And what we are seeing with it is 30, 40, 60, 100 bushel an acre yield increase, which has been dramatic and provides a very strong ROI to corn farmers.